Oh, I did post the other recording. Sorry, somebody finally reminded me. We have a lot of reactions that are spontaneous, like combustion is spontaneous. But when you're sitting in class, you never think, oh, I'm going to catch on fire, right? Because combustion is spontaneous. So if you were to catch on fire, I mean, you would burn down into little ashes. It does happen. In cremation, when we cremate people or have funeral pyres, I mean, people just burn, right? That's a spontaneous reaction. The reason it just doesn't happen all the time is because there's a lot of activation energy. There has to be enough energy to start the reaction, and then once it starts, it keeps itself going by producing more energy. So a lot of reactions have, we say, high activation energy barriers. That's what that is. Okay. That means they're slow. And what an enzyme does is it changes the reaction so it happens with less energy. It doesn't change the amount of product that you get, it just changes how fast that you get there, okay? So you get the same amount when you have a catalyst, but you get there faster. Okay. So just an example, enzymes, there's an enzyme called carbonic anhydrase, and it catalyzes the conversion of carbon dioxide to carbonate in water. So just give you a quick example of what that reaction is. So CO2 and water. I think my wife has my pen, so I'm using this weird thing, so I'm writing funny. Makes HCO3 minus plus H plus. And it's an enzyme. An enzyme's a catalyst. Do you remember where you write the catalyst on the reaction? Above the arrow. So it would be over here, carbonic anhydrase. Okay. Can you read it? Start out really big. You look like first grader writing on the left.
Looks like my homework.
Oh yeah, could be. All right. So enzymes are almost all globular. So this is a little bit of a review. What's a globular enzyme? It's like a blob. Yeah, it's a blobby enzyme. Hydrophilic or hydrophobic on the outside? Hydrophilic on the outside. Hydrophilic because they have to be, they're globular, they're in solution. And if they're going to be in solution, they have to sort of bond with the water. So you need the hydrophilic part on the outside. Yeah, it's the opposite of a transmembrane protein. Transmembranes usually have the hydrophobics on the outside, and then they have ions that can go through the middle of them, so the charged parts are on the inside. So a globular protein, sorry, I started writing something. Polar groups out. Nonpolar in. And that's because they have to be in solution, okay? It's probably the most efficient way for them to get to the substrate, whatever they're reacting with, is to be in the solution. There's a type of enzyme called a transmembrane protein. That's what we were just referring to now in our little conversation up here. Transmembrane protein go through the cell wall, like the cell membrane. So those that usually have the hydrophobic parts on the outside, so they're different than these, okay? But you don't see them freely floating around. They're actually embedded in a membrane. So the thing about an enzyme, it has a couple of different parts. And remember, an enzyme is a protein. So we talk about proteins, right? We have primary structure, secondary, tertiary, quaternary structure, and they can be really big. But in a protein, it usually has an area called an active site. And a lot of times when you see them drawn, I'm going to draw mine like a Pac-Man. A lot of times they draw them upwards. But they're drawn like this, and then this part is the active site. it's so slow. And the active site is usually fairly specific for one type of molecule or one molecule only, and that's known as the substrate. So whatever it reacts on is called the substrate. Gosh. So the substrate goes into that active site, and that's where the reaction takes place. Now, let's say the substrate has an OH group on it, right? So the molecule that's reacting has an OH group on it. In the active site, typically, you'll have another functional group that could hydrogen bond to that. So the active site, this thing that we always draw as like a wedge, like this, or we draw it as a little box or a little circle, right? That actually has functional groups for amino acids on the inside that actually have specific bindings for the substrate. And they're oriented in such a way so that just the substrate can fit, okay? Oh, hang on. I have a stupid example. Sorry. No, I think I'll be okay. This is my enzyme substrate model. Okay, so long time ago, they came up with these ideas about how this works, and I don't know where the slide is on this, so I'm just going to start talking about it now. One mechanism by the way this works is that it fits exactly, like the substrate is an exact fit for the active site. You guys know what that's called? The lock and key. Okay, I will just say this is being replaced by another model, but the lock and key is a great idea because how does a key work, right? You take your, well, I don't have my keys. You take your key and you take it and you stick it in, right? And there's a bunch of little tumbler thingies there, but it's only that key that will fit, hopefully, unless somebody has your keys, will fit and allow you to open the lock, right? So that's one description of how the active site works, is the lock and key kind of thing. The other one is known as an induced fit. So the lock and key mechanism is really old. Remember we did Fisher diagrams? We talked about sugars, the Fisher diagrams of sugars, and there's the left, right, or the right, right, left, right for glucose thing. Who came up with that Fisher diagram? Fisher, yeah. He also came up with the idea of lock and key. This is back hundreds of years ago. Okay, so, well, 150, 160 years. The other one's the induced fit. And I like the induced fit because you think about it, like if this is the enzyme, okay, and then this is the substrate, right? They're not the same shape, right? There's a little bit of, like this is the one size fits all, right? So, well, one size fits large, but you take your hand and you stick it in here. What does the glove do to your hand? Well, it tightens around some spots, right? And the loose spots, so your finger goes in the loose spots and it fits your hand. This is the induced fit. And what the induced fit model is that this pocket isn't exactly the right shape. But when the substrate goes in there, then it clamps down on it, changes the shape of the enzyme. And in part, that induces the reaction to take place. There's a lot of evidence for this kind of behavior because you can take the enzyme with and without the substrate present and take a picture of it called X-ray crystallography. And you can see that the shape changes as it binds to the substrate. So a real lock, right, doesn't change its shape when you stick a key in it. Like you stick the key in it, that's it, right? An induced fit changes the shape, and it kind of makes more sense because molecules are flexible, okay? the kinds of interactions that these either active site or the induced fit use are hydrogen bonds, salt bridges, hydrophobic, hydrophilic interactions. You can get all that by looking at the substrate and then looking where it binds. But like I said, if it has an OH, substrate has an OH on it, it's usually the enzyme, the active site will have something like a hydrogen bonding group in there to help hold on to it. So this is lactase. It catalyzes the reaction of lactose into glucose and galactose. I've always thought this was fascinating. This is the enzyme. It's got 4 subunits. Do you see the active site? It's this tiny thing. Now, some enzymes will have multiple active sites. There'll be 2 or 3 active sites in them, but oftentimes it's just one, and the substrate is just this. And then in that active site, you have a series of other molecules which essentially surround the lactose and hold it in place for the reaction to happen. Okay. Yeah, I think that's crazy. How big of a molecule it is to just do that one reaction. But they're all like that. They're gigantic molecules. Their only purpose is to do something very simple like this. Okay.
Does the enzyme lose an H to make that happen?
What's that?
Does the enzyme lose an H?
No, it uses a water, I think. Yeah, it does a hydrolysis. So it just speeds up the hydrolysis. And I actually think, if I remember right, this one's reversible, so it can actually cause it to split and cause it to form faster. But I don't quite remember all those details. So. Yeah. But that active site just catalyzes that one reaction. So for many reactions, right, in your body, there's just one enzyme that does that. So think about all the reactions in your body. There's lots and lots of enzymes. Then there's some enzymes that essentially do classes of reactions, like do hydrolysis of lipids. So all the lipids get hydrolyzed by one enzyme, that kind of thing. Okay. So in terms of specificity, this is what I was just alluding to now. There's what we call absolute specificity. That's the one substrate, one enzyme model. Okay. Then there's group specificity, so one type of substrate, so one kind of lipid, one kind of, so lipids in general, or alpha 1,4 glycosidic bonds, right? One class. It doesn't care what the things are on either side of the bond. It just cares about that one type of bond. And so we have, that's the linkage specific. And then there's group specificity, so one kind of substrate, carboxylic acids, lipids. They give some examples. Right, urease catalyzes only the hydrolysis of urea, so that's an absolute specificity. Hexokinase adds a phosphate group to hexoses. Doesn't matter what the hexose is, it'll just add phosphate groups to it. Okay. And then linkage, chymotrypsin, and there's several of these kinds of trypsin molecules, but their job is to hydrolyze peptide bonds. So what's a peptide bond? You need to know that. It's the bond between amino acids and a protein. So what these things do will cut proteins up at specific points. Okay. So this may be a little bit more complicated than it needs to be, but think about the process of how an enzyme works. This is just some terminology, okay? You have the enzyme and you have the substrate, and the two come together, and that doesn't necessarily mean the reaction happens, okay? So I'm going to try to explain to you on this diagram what's going on. So this is the enzyme, and that's the substrate. The first thing they do in the course of the reaction is they bind together. So we call this the ES complex, or the enzyme substrate complex. That's this up here. Then what happens is the reaction takes place, so it becomes the enzyme product complex. That's that. So the actual reaction takes place here. And then once the product is formed, because the product is different than the reactant, and remember, the reactant is the substrate. The substrate comes into the active site. Why does it bind there? Well, no, that's what it's supposed, that's the function, but why does the substrate bind to the active site again? Yeah, there's specific interactions with amino acids. So it binds to the active site because of its shape. So once the reaction takes place, what happens to the shape of the molecule? Changes. So then it leaves the active site. So you have a substrate come in, right? Comes into the enzyme, it makes the enzyme substrate, then the reaction takes place, so this thing changes. It's no longer attracted or as attracted to the enzyme anymore, so it floats out, and then another substrate will come in. So you notice what happens in this whole process. You started with an enzyme here, oops, and you ended back with the enzyme here. So the net really is the enzyme reacts with the substrate to produce the product, but the enzyme during the process gets regenerated. Okay? So that's why when we write chemical reactions, like if I were to write this as a chemical reaction, I would put substrate, product, and then enzyme goes here. Okay? Or E. Because it doesn't actually get used during the course of the reaction. Okay. I'm going to skip on this maybe. We'll go over to it. So this is another picture, another way of viewing the lactase enzyme. So here's the enzyme here. Here's the substrate, and because of its shape, it fits into this enzyme and produces the ES, enzyme substrate complex. Okay? Water comes in, and this bond gets hydrolyzed, and now the shape has changed, and so the two molecules leave. These are the product molecules, and the enzyme's free to react again. Okay? Part of this is, I don't know if you noticed, it says a flexible active site in lactase and the flexible substrate lactose adjusts to provide the best fit for the hydrolysis reaction. That's actually following the induced fit model. So that lock and key idea is being replaced more by this induced fit model.
Is there any type of enzymes that change shape or like aren't able to be used because of the complex that's formed? Do you know what I'm like trying to say? Like
Yeah. No.
There's that fit.
By definition, an enzyme, it has to change it such a way that it's reusable. So it forms a product that no longer is attracted and leaves. Now, there are compounds that we call, for example, competitive inhibitors.
Yeah.
Yeah, we'll talk about that. But a competitive inhibitor is something that slows an enzyme reaction down. And the way that usually works is it works by, it's similar to the substrate, and it competes for the active site, and then but then doesn't react.
But that's not changing the shape of the actual enzyme.
No, it's just keeping the substrate from getting in there. So it kind of is changing it because it's like, you know, you're eating food, you have your mouth full, and then you're eating like cake, and then somebody hands you your dinner, you're like, but I'm eating cake, and my mouth full. Can't stick something else in there and make a reaction go unless I spit this thing out. So that's the competitive inhibitor thing, right? Cake fits your mouth, food fits your mouth.
But by definition, it should never change.
No, I don't think it changes in the sense like maybe you're thinking, like it changes and never can be used
Yeah,
again. That's a non-competitive inhibitor. So there's different kinds of, like, that's not, again, that's not an enzyme reaction that changes the shape of the enzyme so it's not usable. That's something that you're putting in there that keeps it from reacting.
Like cooling it.
No,
Slows
so,
it down.
inhibitors are chemical compounds.
Okay.
So, for example, aspirin is a non-competitive inhibitor of this cyclooxygenase, and it actually binds to the active site and changes one of the amino acids in the active sites and keeps the enzyme from doing its thing. But it does that by permanently or semi-permanently altering the active site and keeping something else from getting in there. Okay. If that makes sense. Okay. Well, okay, so this stopped working again. I think our internet is being very finicky. Oh no, it's Windows wanting to update again. Good gosh, keep doing this. Postpone for 4 hours. Let's see if this works. Oh, sorry. I talked about this already. So there's the lock and key and induced fit. So I'm going to skip over this part. Oh, yeah, but the induced fit idea is the substrate in the enzyme adjusts their shapes to make a better fit. So like my hand is fat in some spots, but the tight glove is tight, so it just squeezes in there, right? So my hand is the substrate. So which is the active site? Entire enzyme, section of the enzyme, the substrate? Yeah, it's a section of the enzyme, usually a very tiny part of it, okay? In the induced fit model, what happens to the shape of the enzyme when the substrate binds? Yeah, taps the shape. So like think of a glove. This is the induced fit, okay? Wow, that was not too bad. Next, sorry. That's not what I wanted. I mean, it's what I want. There you go. We're talking about name and classification of enzymes. Okay, so, and I'll go for maybe another, let me go for another 25 minutes or so, and then we'll review in essence. All right, so enzymes, when we name them, we basically name them for the compound or what they do. So the compound they react with or what they do, the type of reaction they do, okay? So alanine transaminase, all right? The trans means it moves an amino group. So we'll see examples, but this is an example of a name. Alanine is the molecule it works on. Transaminase means it's moving an amine group, okay? So that's an example of the name being a composition of the molecule it works with and or the type of reaction that it does. So most of the enzymes end in ASE, okay? And then it identifies the reacting substance or the kind of what the enzyme does. Or there can be common names. And there's a couple of common names that you have to know as enzymes, I think, just for your own benefit. Maybe I'll ask it on a test and it'll be that kind of benefit. But pepsin and trypsin, they end in IN, but those are enzymes. But those are common names of them. Okay? So you have enzymes, okay, and then you have the substrate, and then sometimes there are things which we call cofactors. There's two kinds of cofactors, and basically it boils down to ones that are inorganic, like salts, or like magnesium or iron can be cofactors. Those are inorganic. What do I mean by inorganic? Right? Metals, right? Usually they're metals. Sometimes people refer to them as helper molecules. And then we have coenzymes, and coenzymes are usually organic in nature, so like carbons and hydrogens. And those are actually what we call vitamins. When we're taking a vitamin, actually what you're doing is you're taking a cofactor. It's a type of coenzyme, they call it, but coenzymes are kinds of cofactors. They're used to help organic reactions. Okay? The organic reactions being the enzyme reactions. So these are not proteins, right? They're small molecules, not peptides. So they work with the protein, but are not proteins. We call them helper molecules. If they're organic, we call them vitamins, okay? or coenzymes. Okay, so I'm resuming recording, so maybe this one will work. So I didn't know this actually. I didn't know there was an enzyme commission of the International Union of Biochemistry and Molecular Biology, and they have classified enzymes into 6 general types. So you need to know the 6 general types. There's oxidation reduction enzymes, which we call oxoreductases. That kind of makes sense for oxidation and reduction. And it uses, actually, you have the substrate, if you'll notice here, there's the substrate. It uses a coenzyme, NAD+. And it's an alcohol dehydrogenase. So it causes what happened here in the molecule from here to here. What's that? The OH broke apart, and I formed a double bond here, right? So I removed actually 2 hydrogens. So if I remove hydrogen, what is that? It's an oxidation reaction. So this is an oxidation reaction. So NAD plus is actually the coenzyme that picks up the hydrogens and helps to oxidize the alcohol, the ethyl alcohol, into an ethanol. So this is now aldehyde now. And then produces NADH and an H plus. Okay. So in this case, we talk about cofactors. The cofactor is this NAD plus. There's what we call transferases. So if it's an aminotransferase, it transfers an amino group. If it's a transaminase, it takes the amine. So watch, see if you can follow this. I don't know if it's easy or not. I'm trying to find a place to point. Oh, there we go. So here's my alanine with the NH3 group. Where does that NH3 plus group end up? It ends up replacing that functional group. Okay. So that's a transferase. It assists in moving a functional group. And just as some examples, I don't know that you need to know all these types of examples. A transaminase transfers the amino group from one substrate to another, and it uses actually energy to do that, ATP. We haven't covered that yet, so I don't need to know too much about that yet. Kinases transfer phosphate groups from one substrate to another. So if you have ATP and ADP and all those things that you learn about in biology. And then hydrolases just catalyze hydrolysis. So hydrolase catalyzes hydrolysis. So this is a protease. Proteases catalyze the hydrolysis of peptide bonds in proteins. So this is the peptide bond. And if you stick a water in here, you can get a carboxylic acid and an NH2. But then it's under physiological pHs, it's a carboxylate and the protonated amine terminal. Okay, so that's what a hydrolase does, just catalyzes the hydrolysis. So lyase just cuts things off. Okay, so here is a carboxylate, and pyruvate decarboxylase just cuts this off and replaces it with a hydrogen, and you get CO2 out. But there are non-hydrolysis reactions. So hydrolase cuts it with hydrolysis, and then lyases just cut them, just like pull a group off. Isomerases are interesting. Here is a cis double bond. You'll see this one here. And there's a trans double bond. It does take quite a bit of energy to actually make these happen unless you have an enzyme to do it, okay? And so an isomerase would convert it from being a cis to a trans double bond. And a ligase simply connects two things together. So here's, for example, a CH3, and you'll notice here it added a carboxylate group on the end. It made it into carboxylic acid. Right? It uses energy to do that and then replaces a hydrogen on the CH3 with the CO2 molecule, essentially, and produces a carboxylate. By the way, this, it seems weird, that's a phosphate group. Okay. It's a little bit of, what's that? So we took an ATP. Yeah, so this is an ATP. We haven't learned about that yet, but we'll cover that in a later section. This is an ATP. ATP is a high energy molecule. It stores energy in phosphate bonds. And then when that bond breaks, it uses that energy to convert this to a carboxylate. The energy goes when this ATP is adenosine triphosphate and ADP is adenosine diphosphate. And when you lose that one phosphate group, that's where you get the energy from. This is actually like a phosphate. It's not phosphorus, I don't think. And then a hydrogen. So, oops. So an aminase does what? What is aminase? Sounds like an amine, right? So what do you think it is? Adds an NH3 plus, right? Dehydrogenase? What's that? B, removes hydrogen as atoms to form double bonds. Well, and a synthetase makes synthesis, makes new things, right? So it's C, combines two molecules to make new. So you have to, when you look at the names, you can kind of guess at what it's doing, right? This is the whole point of the slide. You don't know a lot of these, right? But if you break down the name, like isomerase, makes isomers, right? Changes isomers, so then you have to think it's cis and trans. So that's A, okay? Synthetase means literally to make new. So you're going to make a new compound out of that. So that would be C. Oh, I'm going to skip this one. As fun as it might be. Let me go over one more section. So we talk about this thing called enzyme activity, and we talk about it, we kind of assume everybody knows what it means, okay? So I wrote a simple definition. Enzyme activity is how fast an enzyme catalyzes its reaction, okay? So activity is how fast the reaction goes with the enzyme, okay? So one of the big deals in biochemistry is understanding the factors that affect how active an enzyme is, how fast it can make its reaction go. So a really active enzyme will have a high activity, right? And a really low activity enzyme would be really slow, okay? So there are four factors, I think. I got three up there. But temperature, ion concentration, concentration of enzyme and substrate. I'll talk about all these things as we go along. So first of all, get the idea that enzymes usually have some optimum temperature. In humans, that optimum temperature is usually around 37 degrees Celsius. So it turns out when you have really low temperatures, right, then there's not enough energy for even an enzyme to catalyze a reaction. So at low temperatures, reaction rates always tend to be low. So if the optimum temperature is here, anything lower than the optimum temperature produces slower reactions, and we get that by looking at the reaction rate, how fast the reaction is going here. So this is maximum enzyme activity. That simply means that's the fastest point in the reaction. The reaction goes the fastest there. As the temperature goes lower, they usually have this sort of bell-shaped curve appearance, okay? And this is simply because you have less energy in the system, so the reactions have to go slower. Make sense? Now, if you go to temperatures above the maximum temperature optimum or temperature max, then what happens is the enzyme rate begins to slow down, and it does that because the enzyme begins to denature. So as you put enough energy, remember we talked about proteins and how they're held together by intermolecular forces. If the temperature gets high enough, then the enzyme begins to lose its shape, and then so it begins to lose its activity, okay? So it turns out if you go this way, usually, this tends to be reversible, that you can go up and down in temperature a little bit and the activity stays about the same. But if you go the other way and you denature the protein, that's not reversible. So you can take it and make it hot, and then when you cool it back down, you don't usually have activity. Okay. So there are what we call extremophiles. Thermophile is a type of extremophile, okay? Acidophiles is a type of extremophile. They live under conditions where we would not. So a thermophile can live at temperatures as high as 120 degrees Celsius. Their enzymes are built robustly enough that when you get to those high temperatures, they don't denature. Okay. Stronger intermolecular forces, you could presume they have stronger intermolecular forces holding them together. There's also acidophiles, things that live under very acidic conditions. So like in mines, a lot of times water washes down to the bottom of the mine and goes through sulfur and a bunch of other things. And at the bottom of the mine, you'll find like concentrated sulfuric acid. And even under those conditions, sometimes they find things that are living. Okay. Okay. So, pH, very similar. Right. Most enzymes have a optimum pH. That's where the enzyme operates the fastest. Why do you think our enzymes are typically around 7? They're not all at 7. Some of them are much higher, but that's yeah. Our blood, our bodies are around pH 7, so the maximum activity is typically around pH 7, although it's not always that. Okay. And you have to think about, well, why does this kind of thing happen? Well, the functional groups in the active site typically are pH sensitive, and so by changing the pH, you change its activity. Okay. So again, there's a certain range of pHs where everything's happy. If you take it to pH that's too high, there's not as many hydrogens. If you take it to pH that's low, there's too many hydrogens. That changes the active site and changes the ability for the enzyme to function. Yeah, so there's a couple of interesting optimum pHs for selected enzymes. So pepsin, notice its pH. What's that pH? 1.5 to 2, right? Where does it operate? In your stomach, and your pH in your stomach is around 2, right? So it would be bad if its pH optimum was at 7 because your body would secrete it and then it would just be inactive. Let's see what else. But everything else, typically, I was actually trying to find like why arginase's pH, its optimum pH was so high. I never found anything. But everything else is ballpark around 7. Okay. It doesn't mean your liver is at that pH, but that for some reason it has that high pH optimum. I don't know what it is.
I know. Isn't our liver, I didn't think it was basic like that.
Yeah, I don't know. So I don't know why that has a high optimum pH. It's just not operating. So I tried to look it up, but I didn't find anything. I find all kinds of other things I don't want to read, pretty much. Okay, so this is a little bit of a complicated idea. So if you take, this part makes sense. If you take the enzyme concentration and you increase it, okay, the reaction goes faster. Make sense? More enzyme around, reaction gets to go faster. So if you try to do this graph with the substrate, it looks very different, okay? So, but basically, enzyme activity, the activity of a solution in enzyme will just go up linearly with concentration. When you try to do that with substrate concentration, it levels out like this. Okay, so let me kind of explain to you try to explain to you what's going on. Well, I think I'm gonna have to make a new graph for this. Okay, so let's say my enzyme is going to be a square. Okay, so there's my enzyme, and what I said, and then I have substrate, and so when the substrate goes through here at very low substrate concentrations, the enzyme can keep up. A substrate comes in, product comes out. That's supposed to be a triangle. Product comes, and product's a triangle, okay? So this guy has to wait for, this one has to wait for this one to be done in order to go through. Now, if the enzyme's really fast, he doesn't have to wait very long, so as you increase the concentration, right, you increase the rate. So let's back this up again. Let me make my diagram like this. So let's say this is the situation like this. If the enzyme is fast enough, if I add a third one over here, right, the reaction will be faster. So instead of being one going through, I could have all three going through if the enzyme is fast enough. But at some point, if I add more and more and more, some of those substrates will be sitting around waiting for a free enzyme to go through, okay? And once it goes through, right, then the reaction, once the one before it goes through, so once this one goes through, then this one can go through. So what that looks like on a graph is something like this. Here's the substrate concentration. Here's the rate. We often refer to that as the velocity. Because what is when you're driving your car and you're going miles per hour, right? Or you could do moles per second, it's a rate. So we call that a velocity. If the substrate constant, and this is for constant enzyme concentration, the graph looks like this. It just goes up linearly. So increase substrate, rate goes higher. Increase substrate concentration, rate goes higher. At some point, right, the substrates can't find a free enzyme to bind to. All the enzymes are busy trying to do another reaction. And so what happens is as the substrate concentration goes up, the rate of the reaction doesn't go up. And eventually it flattens out like this. Okay? If I added more enzyme to the system, then that plateau would be higher up. So for more enzyme, we change the color. Let's do that. If I added more enzyme to it, it would do this. Different maximum velocity, okay, for different maximum enzyme. But in essence, though, all of them behave the same. That is, as I increase the concentration, it just doesn't go up forever. It has to be limited by the amount of enzyme that's available to do the reaction. So, let's see. Back to this chart. That's what this chart is here. This is the maximum activity. We often refer to it as the maximum velocity. To sort of tie into the theme that we have of it's like how fast you're driving your car. And that's really related. Vmax is what we like to call it as well, because we hate saying velocity all the time. Okay. Vmax is limited by the concentration of enzyme, and you can't just keep shoving substrate into the enzyme. Eventually, it has to max out at some point. And this plateau here, that they point out, is when you finally filled up all the enzymes with substrate, and there's just substrate waiting around to react. Okay. So let's go over this. You have three choices for the answers. 1, 2, or 3, no change, increase, and decrease. And it says sucrase has an optimum temperature of, what do you think sucrase does? Breaks apart sucrose into glucose and fructose, right? Has optimum temperature of 37 and an optimum pH of 6.2. If you increase the concentration of sucrase, what does it have? What effect does it have on the rate of the reaction? Goes up, right? If you change the pH to 4, what happens to the rate of the reaction? Decrease. Run it at 70? Decrease. Yeah, everything goes down from the optimum. Yeah.
It goes down on C?
Yeah. Because its optimum is for C, the optimum temperature is 37, right? So if you go to a higher temperature, it's no longer at its optimum. That's where it's the fastest. So anywhere off the optimum temperature is slower. So the rate goes down. What happens if I increase the concentration of substrate? Not a simple answer.
Increase first and then
Increases first, plateaus eventually, plateaus, okay? So it depends on where you are on the, what we call the Vmax curve, right? Wherever you are on that Vmax curve, if you're at the low end of it, you just go up, you go faster. If you're at the high end of it, then you just stay at the constant rate, no change. All right, good. Let's go over exams material. Do you have a book I can borrow? It'll work. I didn't want to say I loaned my book out, nobody gave it back to me. I told you to keep it. I could pull up the ebook, but that's no fun. All right, let me find out where we're at in this book. So we are doing carbohydrates, right? Lipids and amino acids and proteins. So that's starting at 15. All right, we kind of beat 15 to death, if you ask me, but
That's what we quizzed on, right?
yeah, that's what we quizzed on. Same, expect the same kind of material. Okay, so let's do this old school review style. Stop the recording though.